Welcome to the Department of Clinical Dentistry. Follow me to the Tissue Engineering Lab. And now, a few of our PhD students are going to show you how we use stem cells to create bone in the lab. But before we get in, we need to get our lab coats on. This is our cell culture lab, where we isolate stem cells, and then it's very important that the environment stays sterile. That's why I have to change my lab coat. Hi, Sameh.
Hi Salwa.
What are you doing today?
Today we got a bone marrow sample, and you know what? Bone marrow is a very common source for stem cells.
Can you show us how you isolate stem cells?
Yes, of course. Follow me, please. First, I filter the bone marrow from any blood clots. Now, we collect the cells from the bone marrow. After we collected the cells, we add nutrients to the cells before growing them in flasks.
Because the number of stem cells in the bone marrow is very few, we need to expand them before we can put them back inside the body. Right now, I will be changing the medium to give the cells fresh nutrients to expand.
Can I see the stem cells now?
Yes, sure. Ayla will show you.
This is incubator where we keep our stem cells under 37 degrees. Now I will show how stem cells look like under the microscope. This is how the stem cells look like under the microscope when they are attached to the plastic flask. You can see. And now it looks like that we have a good number of cells, and we need to prepare the carrier in order to implant these cells into the body.
I'm scanning bone by using micro-CT scanner to design stem cell carriers we call bone scaffold with the exact shape of the bone. And this is how scanned bone tissue looks like.
Hi, Maria. Can you show us what you're preparing?
Yes, I can. I am now preparing a gel material, as you can see here, that will be used as an ink to 3D print the stem cells.
Now I'm going to collect the stem cells and mix them together with the gel material to form the bioink.
Now come and join me. I'm going to show you how I print stem cells in the gel bioink to create a proper environment for the cells to grow and form bone. But because this bioink gel is mechanically weak, we need this 3D printed scaffold to give the gel enough strength to be implanted in the bone.
So here I am in front of the mechanical testing machine, and now thanks to this machine I will be able to assess the mechanical strength of scaffold construct for in vivo implantation of the constructs. So thanks to the data we obtain, we can compare the mechanical strength of the scaffold construct with mechanical strength of actual real bone.
I'm working with a machine called the bioreactor, where we can grow stem cells on the scaffold in an environment where nutrients is floating like blood in our body. This is the nutrients which go through tubes like blood vessel in our body towards the stem cells loaded on the scaffold.
Hi, Francesco.
Hi. Hey, welcome to the microscopy room. Here we are trying to analyze the stem cell scaffold construct that we have seen growing in the bioreactor before. The picture that you can see here next to me is the same coming from the high-resolution microscope, and the sample is the same, and here we are trying to analyze if those cells are producing all the elements of the matrix that one day will become bone.
Hi Neha, can you show us what you're doing?
Hi, so before we implant the stem cell and scaffold construct into the human body, we have to make sure that it's safe, and for that we do this implantation into the animals first. And here I'm looking into this section with implanted stem cell and scaffold construct, and we can see that this pink area represents the newly formed bone. Then I can also stain this tissue with different type of special stains to identify the immune cells, and here we can identify two different type of immune cells with red and green stains.
This was the last station in our short tour.
I hope you learned a little bit more about stem cells and bone regeneration. Now it's time for you to get to know our scientists.
My name is Sameh Mohammed Ahmed. I have a background as a dentist from Sudan. I am a postdoc here at the Tissue Engineering Group. When I was working in Sudan in the Department of Surgery, it was difficult for me to see those patients with massive bone loss. And the available treatments at that time were not enough to treat those patients. So I joined the Tissue Engineering Group and worked with stem cells, hoping that one day these stem cells can be used as a better treatment for those patients and improve the quality of life of those patients with bone loss.
My name is Nora and I'm from Germany and I studied molecular biology. Now I'm a first-year PhD student and I chose to do a PhD because I believe it will provide me with the skills, knowledge and competence that I still need to essentially finish my scientific education. And then after the PhD, I will be a professional scientist and fully able to conduct good biomedical research.
I am Eylem from Turkey. I am stem cell biologist and first year PhD student. I continue to my research in stem cell science because I think that is the promising field in science that can cure so many different diseases.
Hi, my name is Serhat. I am from Turkey. I'm chemical engineer and material scientist. Right now, I pursue my PhD in here. I like solving problems and building structures and drawing and contributing to scientific field motivates me.
My name is Marie Constanze and I come from Norway. This is my first year in my PhD and I have a background in molecular biology, and the reason I chose this field is because of my big interest in biology.
My name is Natalia. I am from Colombia and I am a biologist. I'm also a first-year PhD student. I like to do science because I love to know how the body works. And working with the stem cells and tissue engineering and tissue regeneration is one way to do it.
I'm Masume. I studied chemical engineering in Iran, and now I'm a first-year PhD student here. And what fascinates me about PhD and science is that it keeps the creativity and curiosity on.
So my name is Theo. I am a chemical engineer from France, and I came here to start a PhD in tissue engineering. The reason why I decided to start to study this field, tissue engineering, is because I'm convinced that this research field that is at the crossroad between biology, chemistry and engineering will lead to incredible discoveries.
Hi, I'm Shantaro Yamada. I'm originally from Japan. I have the background as a dentist, but also I've started my research work a few years ago. When I changed my career from clinician to the researcher, actually my mentor told me that the researcher is a producer of evidence, while the clinician is a consumer of evidence. That drove me and then brought me into the research field.
My name is Francesco. I'm from Italy. My background is in dentistry, and I have a Bachelor of Dental Science and a Master of Science in Stem Cell Biology and Regenerative Dentistry. And now I'm a first-year PhD Research Fellow here at UIB, and what fascinates me about science is the multidisciplinarity of our project. Here I have the chance to work with people from many different backgrounds, like chemical engineers, material scientists, stem cell biologists, and signaling experts. And overall, the technology we are developing in here could be beneficial for end users and patients, and for creating value for the society as a whole.
Hi everyone, my name is Neha and I'm a final year PhD candidate here in the group. I come from India and my background is Masters in Biochemistry. Why I wanted to do research is because I was always passionate about human biology from my childhood days, and I think now it gives me a very exciting opportunity to be at the edge of novel inventions and to contribute to the better health and safety of our society.